The overall goal of this procedure is to create a system that lacks cerebrospinal fluid, or CSF, within the embryonic brain ventricular system. This is accomplished by first manually draining embryonic CSF, or ECSF, from the zebrafish brain ventricles. Next, the ECSF is collected for further analysis of content. Then, potential factors are reintroduced into the drained brain ventricles. Finally, the effective removal of ECSF and reintroduction of factors on brain development is analyzed. Ultimately, the function of zebrafish ECSF can be shown through monitoring aspects of brain development such as cell division, death, and neurogenesis. Additionally, ECSF content can be determined by mass spectrometry.
Using this technique, we can begin to study the function of embryonic cerebrospinal fluid during development. It can also be applied to other systems to further understand diseases which alter CSF composition and volume, such as hydrocephalus.
To begin, fill an oil injector apparatus with mineral oil according to the manufacturer's instructions. After pulling microinjection needles, mount a needle on the micromanipulator connected to the oil injector. Carefully break the tip of the needle. For uniform size, measure with a micrometer or compare to a reference needle. Fill the needle with oil using the oil injector to push oil down the length.